In previous lectures, I have several times mentioned the importance of genetic structure, and this is the topic of this lecture. Before we can go into what is genetic structure, we will also need to make a few definitions. So we say that a population has a structure when there are large-scale systematic differences in ancestry, or if we see groups of people who share more recent ancestry. And then the question is, how can we characterize and quantify this ancestry? So we will start with a few definitions, and one definition which will be very, very important and useful later on is the definition of identity by descent. So we say that two alleles are identical by descent when they are copies of exactly the same ancestral allele. And now let's get to the structure, and one of the simplest examples of structure is related to inbreeding. So on the picture you can see an inbred pedigree. So the individual Jai is an offspring of inbred family. So his parents or her parents are the cousins. And now you can see that if we try to trace the ancestral alleles, so alleles which come from the grandparents of the individual Jai, then there is a chance, there is a possibility that the individual Jai receives exactly the same copy of the same ancestral allele, right? So these will be alleles identical by descent, but now that the event is that these two alleles come into, homozygote in the same individual, we can also call this homozygosity by descent or autozygosity. So for this specific pedigree, let's think what are the chances of homozygosity by descent for this individual at some random location in the genome. So we can track this red allele, which is depicted on the figure, and we can see that the chance that this specific red allele is transmitted to individual D is 50%. And this is the same chance, 50%, that exactly this red allele will be transmitted to individual E, and so on and so forth. So for this red allele, the chance that it comes into homozygous form, into homozygous by descent form in individual Jai is 1/64. But in principle, when we talk about inbreeding or homozygosity by descent, normally we are not interested in specific allele, the red allele, for example. And then if we think of the chances of homozygosity by descent for any allele, then okay, we have 1/64 chance for the red ancestral allele, to be homozygous in individual Jai, but you see other alleles, the blue and the yellow and the green, right? So in principle, there are 4 ancestral alleles which could become homozygous by descent in individual Jai. So the total chance is 4 times 1/64. So given a pedigree, you can easily compute coefficient of inbreeding for any person, and you can characterize with coefficient of inbreeding a specific person from specific pedigree, or actually this is also characteristics for your population, because if in population there is a systematic inbreeding, right, this average inbreeding level will tell you something important about the breeding structure of this specific population. Then coefficient of inbreeding tells you about genetic structure in terms of one person, right? But sometimes, and very frequently actually, we are interested about a relationship, the structure between genetic structure which relates to individuals. And in this case, we use a coefficient of kinship. So coefficient of kinship is defined for a couple of individuals. We can call them I and J, and this is the chance, the probability that two alleles at the same locus, random locus, sampled at random, are identical by descent. And what is important to realize, if we take two individuals which are coming from outbred population, so there are no loops, there is no inbreeding in this pedigree, then coefficient of kinship between these two individuals is 0. And if we consider individuals from inbred lines, so there is a very, very inbred pedigree on the top of these individuals, their coefficient of kinship could go as high as unity, right? This is not something which happens in humans, but this is something which is happening for inbred mouse lines or could happen for some plants. There is a relation between coefficient of kinship and coefficient of inbreeding, and basically the coefficient of kinship between individual I and J is the same as coefficient of inbreeding for a hypothetical offspring of I and J. Other measure which is frequently used to measure relationship between people is coefficient of relationship, which is defined as twice the coefficient of kinship. And in case that you are considering individuals from outbred populations, there is a very important physical interpretation of coefficient of relationship, which is basically the proportion of the genome these individuals are expected to share identical by descent. Let us consider a specific example. On the figure you see an example pedigree for which we can compute the coefficient of relationship, right? So we can compute a pairwise coefficient of relationship, the coefficient of relationship between each pair of individuals in this pedigree. So remember, it's defined as expected proportion of the genome shared IBD. So it's, kind of easy to estimate that for SIPs, you expect 50% of their genome shared IBD, or for half SIPs, like the individuals 1 and 2 from the picture, this proportion is quarter, and so forth, so on and so forth. You can compute these pairwise coefficients of relationship, and important presentation of this coefficient is in a presentation in the matrix form, which you can also see on the figure. So from this matrix, we can immediately see what is the coefficient of relationship for any specific pair of individuals in this pedigree. Other interesting thing about coefficient of relationship is that in principle, when you have pedigree, it's relatively easy to compute the coefficient of relationship, this expected proportion of genome shared identical by descent. But in an era of genomic studies, you actually do not need to know the pedigree, because you can simply look into the genomes, and the correlation along your genomes can tell you about the relationship and kinship between individuals. On the screen, you can see the expression which allows you to compute, to estimate the coefficient of relationship between any two pairs, any pair of individuals in question, if you do have genome-wide data for these people. One important thing to realize when you do estimate the coefficient of relationship using genomic data is that you do use frequencies of alleles in the formula which I've been just demonstrating. And then, strictly speaking, for this estimate of relationship to be correct, you need to be using the frequencies of alleles which come from the founder population, which you don't observe. You observe only the individuals, the samples you're working with. And in that, these estimates may be biased, but for most of the practical applications, using the allelic frequencies directly from your sample may work pretty well, as soon as you're working within one distinct genetic population. Next question is, if we know relationship between individuals, can we tell something about correlation of their phenotypes? So what is the resemblance between relatives? In case we assume a polygenic model, and when I'm saying we assume polygenic model, this is to say that the trait, the phenotype we are considering is under the control of really many alleles, many genes which are spread throughout the genome. So if we consider a polygenically controlled trait, then actually the coefficient of relationship will be very helpful in trying to figure out what is the resemblance between relatives. Because if we imagine a trait which is totally controlled by genes, right, by the genome, then we would expect that the correlation between the phenotypes of the relatives is exactly as correlation between their genomes. But we already defined that correlation between their genomes is the relationship coefficient. So in that, if we consider a trait which is only under control of genes, then for monozygotic twins, we would expect that their phenotypes are exactly the same. So the correlation is 1. If we consider SIPs, they share 50% of their genome, so we would expect correlation of 50%, and this is exactly the relationship coefficient between them. However, well, any trait, especially the complex traits, polygenic traits, are both under the control of genome and the environment, and in that only the proportion of the trait's variance, which is under control of the genome, is the one which defines the resemblance between relatives. So in that, the correlation between phenotypes of relatives should be proportional to their relationship coefficients, but there should be some scaling factor, right? So the more is the effect of environment, the smaller will be these coefficients, and the faster the resemblance between relatives falls. So this scaling coefficient is known as coefficient of heritability, and it's defined as the proportion of the trait's variance which is under the control of additive genetic factors.